Okay, just because this classification and regression tree lab is actually not 100% trivial, I will give you a walkthrough and interpretation as well, but definitely give it a shot yourself first. So work your way through this lab and then afterwards watch this video and see what you figured out and what your thoughts match my interpretation that I add here. Okay, so let's start with the lab, and one of the first things you might encounter is if you try to install this package here, mvpart, and you're using a reasonably updated version. So we're trying to install this here, and we get a warning message, mvpart is not available for R, version 4.02. That's an error that you encounter really frequently, and that doesn't mean the package is bad or outdated. It's just that the author didn't bother to upgrade it to the latest version of R. So you almost always have to work with multiple versions of R. So one thing you can do is just install an older version of R and then find the old binaries. So what you can do is search for mvpart binaries in this particular case if you want to install an older package. Let's do this. And there's actually, Microsoft maintains a very good set of binaries, so for Windows, for Mac OS as well. And you can download and install these zip files manually from the menu here. So you have to run an older version of R. You have to check when these were published, so 2014, that's a while back, and pick an R version that's compatible with that. And then you can here install packages from local files. So your downloaded zip file, you can navigate to this, and that does it. Now, another thing you can do is you can install R tools. So this will allow you to compile that package for the version that you're using. I find that very convenient. So you have to once go to this website, and if you're on Windows, and install R tools. So that's just R tools. You download that executable here for Windows 64-bit in most cases. And you don't actually need to do this for Mac and Linux. They already have a compiler built in. So only for Windows this step is necessary. And then you can install developer tools, and then you use these to install older packages. So you can install virtually anything with this command here, install from GitHub, and the particular package that you want. So that almost always works, even though you might encounter bugs, but I never encountered any bugs. So that way you can have your old R packages run on your latest versions. So let's see how this works. So we're working again with the Alberta plot data set that we are familiar with. So we have a bunch of plots that come from particular locations. Near weather stations in this particular case. So I have climate variables and I have frequencies, and I prepared a data set where we have frequencies in percent and also presence absence. So whenever this is not a 0, I'm just calling this a 1. So just to demonstrate two versions of classification and regression trees. So I'll attach it to make the code a little simpler, and we can call the library that we previously installed. And so that mvpart function, that is a multivariate regression tree, but it also works with a single response variable. So we can just do one species. So in this case we use paper birch, and we use mean annual temperature, mean warmest month temperature, coldest month temperature, precipitation, and summer precipitation as predictors. So that's the data set that we are using. This one here lets us pick the number of trees, and all leaves equals true gives us all the leaf plots at every node. So let's run this. So because I specified the XVP, I have to pick something. So you can pick between the orange and the red one. So this one here is X value relative error. But the green line here, that is a variance explained. So you see the first, second, third, fourth split, they explain quite a bit of the variance, and then it kind of tapers off. So we can click on this one here just to keep it simple, and then we get our regression tree with 4 groups. And interpretation is pretty straightforward. So our paper birch here likes cold temperatures, so it's a northern species. If the mean coldest month's temperature are really harsh, minus 30 degrees, that's where the species does particularly well. And then under the warmest climate here, it's virtually absent, and under intermediate climate, it has intermediate frequency, so 0.6 and 2.9%. All right, let's try the classification one. So in this case, we define our response variable here as a factor variable. So we're using the presence-absence dataset, but define it as a factor so that MVPart understands that we want to run a classification and not a regression. I could also call this P and A for presence and absence, but since I put them as 0 and 1s, I have to actually define this as a factor. Okay, and so let's run this, pick 3, and now we get the frequency diagrams here. So subalpine fir actually likes high precipitation. So the light blue are the absence samples, the dark blue are the presence samples, and so if we follow the high precipitation and the relatively warm temperature, so above 0 degree, then I end up at high frequencies, and high precipitation, low temperature, that's the alpine, so it doesn't like that. And low precipitation is pretty much everywhere else in the province. That's where it also doesn't occur very frequently. So let's also look at the summary statistics here. I forgot to do it for the last one, but we can look at it here. So these are my splits, 0, 1, and 2, and it gives me the residual error. So here my residual error is 1, everything. After my first split, I explained 50% of the variance, so 50% is the residual error, and then in my second split, I explained about 14% of the variance, so 36 is the residual. And that 36 here is also reported here as an error, so that's the variance that's not explained. Now, I do want to scroll back up and also look at the summary statistic of the previous run here. So let's quickly rerun this here with 4, and let's look at the summary statistic. So there's something interesting here. What it does is it reports the variance after the first split, so that's 35% of the variance, but then it jumps to the third split, so it goes right over this one and jumps to the last one here. And that happens if I explain a bigger variance subsequent to a smaller variance. And that is actually one of the key features of the MVPart package. It looks ahead. It's not simply a recursive algorithm like I explained in the previous video. It actually looks one ahead and sees if it can explain even more variance. And these are interaction variances. So if something is colder than -25 and you also consider the mean warmest month temperature, then it makes a big difference. So that ability to look ahead, that is unique to this package. That's why I like it, and I go through the trouble of installing it with a slightly convoluted procedure. So it gives you the ability to find these interactions. So sometimes you have to split, and then you can come up with homogeneous group based on a second variable. So that's a really cool feature that's unique to the MVPart and the RPart algorithms. And it also provides alternative splits. So in this case, I can use mean annual precipitation as my first split. Mean summer precipitation would actually give me a very similar result, so almost as good. So the higher this improvement value is, the better it is. And even mean warmest month temperature could be used in this particular case. So you see, if you have autocorrelated climate variables, it picks the best one, but there are others that are also relevant. So reporting these is important. So I usually provide a separate table that reports the alternative splits for my first node. And then if we go to our second node, it's again the same 3 variables that you could use to make those splits. So the message really is that certain combinations of precipitation, mean annual temperature, and mean warmest temperature define the climatic niche of that particular species. So you would miss that if you don't look at the alternative splits. All right, and let's try the multivariate extension here. So I have a slightly different data set for that. So I have 5 species now, just the frequencies, and it's already normalized, so everything is expressed standard deviations from a mean of 1. So 0 values actually become a negative value. And my predictor variables are the same. And I also have the ecosystems where those come from, those sample plots. And for this one, I actually have to bring all the species out as a separate matrix. So I have just my species in the data set, spec2, and so that I place as dependent variable. You remember before we just had a single dependent variable, but now we have the whole community as dependent variable. And I'm running the same thing. So lots of option here. I'm gonna pick something that's not too complicated. Let's start with 5 just for first interpretation here. And now I get my frequencies in these leaf plots here for 6 species, so one after the other. From top to bottom is from left to right. And let's check for our extreme environments. So mean coldest month temperature warm goes this way, mean annual temperature warm goes this way. So the warmest environments none of our tree species like, so this is approaching the grasslands. And let's go the opposite, so cold to the right, cold further to the right. So the species that like this is the 3rd, 4th, and 6th. So lodgepole pine, white spruce, and jack pine. These are high elevation or northern, relatively northern species. So and the species that shines at intermediate temperatures here is Populus tremuluides, aspen. And if we go to these ones here, so these don't have extremely harsh winters, but they are still reasonably cold. So mean annual temperature above 1.3, that's my subalpine. And so I get subalpine fir and Engelmann spruce there. And then when it goes to colder temperature yet, then we arrive in the alpine, and none of my species is really frequent there. So I have now a number of groups, and I can attach those to my data file. So oftentimes I want to know which of my plots fall actually into these 1, 2, 3, 4, 5 groups. So to do this, we get the group out of that output file here. And so let's take a look at this. Just bind it to our data file, and we now have, for each of the plots, we have a group. And so my group number 5 is the alpine, as a montane biome, is the alpine. And the way they are numbered, so this is number 5. Let's see if that checks out. So you start over here. This is your first group, second group, third group, fourth group, fifth group. So we identified that as alpine before. So it got that actually correct just based on the frequencies. So this would be 6, this would be 7, this would be 8, this would be 9. So 9 is the harshest, coldest, and most northern climate. So if we go back to our data table here and see where the 9s are, they're right here. So this is a boreal subarctic. So these are in the north and very cold. So again, it got that right. But we only have 5 groups, so there is also a 9 here for the lower boreal highlands. And let's see where else. This one is a 9. So the northern mixedwood, that's also northern boreal, where that got assigned to this group. So we don't have quite enough groups to actually reflect our mapped ecosystems. So let's up this a little bit so we can run this again and maybe pick 10, which is actually suggested here, and we get a more complex diagram, and you have to spend some time thinking your way through all this, but it should make sense and conform to expectations. And now we could actually use this regression tree to make predictions. So we could imagine that we have a new sample point and we want to predict, just based on climate, what kind of species frequencies we would have. So if I have a new sample point with a mean coldest month temperature of -12, I go left, and then I look up what mean annual temperature this has, and I have to go right and left and so on and so forth to arrive at my predictions of my species frequencies. So let's see if we can run those predictions. And in order to merge my predictions in here, so what we're going to do is we're calculating average predictions for our species. So these are my group numbers, and these are my average species frequencies for each of them, because in the end I do just predict the group membership, and then I have to figure out what the average species frequencies are for each of those groups. And now I could do some wall-to-wall projections for climate of Alberta. So if we look at a gridded data set, so that is 40,000 rows long. So for every grid point in Alberta, we have a bunch of climate variables here. So we can now use the output file that has the regression tree recorded and just go through it, go left and right and left and right, depending on the variables, to make our predictions. And so these are our predictions for every single point in my climate data set. Now I don't know where those grids are. I lost this information, so I have to merge this back in. So the order doesn't change. I can just take the X, Y, and ID coordinates and bind them together. So that way I have my grid ID, my X and Y coordinates, and then my predicted species frequencies. And I can also merge my group in if I want to see my multivariate regression tree group, which is my own custom ecosystem, if you will. So now I also have the group attached at the end. So we can look at this with the lattice package. I just use this as a GIS replacement here for simplicity. And there's my habitat for subalpine fir. So indeed it occurs in the subalpine. Jack pine should be quite far north, and sure it is. And Populus tremuloides is kind of intermediate, especially in the dry mixed woods and the parklands. That's where aspen is most frequent. So and central mixed wood as well at somewhat lower frequency. And I can also look at my classes where they would come out, and that is remarkably similar to our actual official ecosystem classification system. So we have the foothills here, the upper foothills, the subalpine, grasslands, parklands, central mixed wood, northern mixed wood, boreal highlands, and subarctic. So this checks out. These are actually the upper boreal highlands and the lower boreal highlands. So it all gets this from just 100 samples throughout the area here. So it's a pretty powerful technique for delineating ecosystems if you have both frequency data and environmental data that you can combine in this way. So, and to round it off, let's take a quick look at randomForest. So this is the same idea, but it's a bootstrapped version that subsamples your data set and uses different predictors and different numbers of rows every time to build regression trees. So that's called randomForest. It's also very widely used for predictive tasks. So we work again with our normal Alberta tree plots, and we're just going to do a prediction here for Abies lasiocarpa. So let's run this one here with 100 regression trees and do the same predictions for climate variables. And you can see you get a bit more definition here, so that's a better representation of where the species is actually occurring. So the first one that we had was a little simplistic. So this randomForest version of regression trees, it does get you much better predictions. So we can also run this for the 2050s. Let's see what happens as climate changes. We would expect this species that's in the upper foothills and subalpine to migrate towards the Rockies, so to high elevations. So let's make the predictions for the 2050s climate and do a plot of that. You can see it shifts to high elevation with the highest frequencies and get lower frequencies here in the lower foothills. So this is just one set of examples how you can use regression trees analysis with two data sets, but they are extremely widely applicable. So originally this one was not developed for biology or ecology. This is a method that has been developed for medicine. So one of the big applications of the randomForest methodology is to predict disease incidence as a function of a whole number of risk factors that come in all kinds of shapes. So it could be smoking, yes or no. Then the next variable may be body mass index, so continuous variable. Third variable may be male or female. Next variable may be age, continuous again, and so on and so forth. So you can line up all kinds of risk factors that may also interact with each other and can make very accurate predictions. For example, what the probability are to get a lung cancer or a heart attack or something like that. So very nice, powerful techniques that can be applied in an enormous amount of different fields. So with that, we really have filled out our standard toolbox, and the rest of the videos are on-demand or volunteer student lectures. So thank you for watching the standard program so far.